American foulbrood is the most devastating brood disease that honeybee colonies get. Unlike the name suggests, American foulbrood doesn't necessarily come from the Americas. It was just simply described there first, similar to the explanation for European foulbrood, which is also not necessarily from Europe, but was just described there first. American foulbrood is the condition caused by the bacterium Paenibacillus larvae. American foulbrood comes in two stages. There's the vegetative stage that causes all of the problems that you see with American foulbrood, and then there's the spore stage that makes American foulbrood persist. So let's talk about this briefly. Larval honeybees become contaminated with American foulbrood bacterium when they eat contaminated food. But unlike European foulbrood that tends to kill the larval stage and maybe into the prepupal stage, American foulbrood tends to kill the prepupal stage or older. So it's the larvae that get infected, but the prepupae or the young pupae that actually die from the disease. What makes it so bad is that the spore stage of the bacterium is incredibly resilient in the environment. It's been shown to be able to survive for decades. So you can do stuff to your colonies to get rid of the vegetative stage so the colony looks healthy and life seems good. But when the colony is in that same box 5 years, 10 years, 15 years, maybe 20 years later, the disease can pop back up. Once the spores are in equipment, they're in equipment. Because of the spore stage and that persistence in hive equipment, and potentially even the bees that are in the nest, there's a series of recommendations built around American foulbrood that are somewhat unique to American foulbrood. I'll talk about that momentarily when I get to the treatment part. So you've got this vegetative bacterium developing in the midgut of the individual bee, the infected bee. How does she present clinical signs of infection? Well, before we try to go into the clinical signs of infection, it's always better to understand how healthy individuals look so that you can compare the diseased individual to the healthy individual. And of course, larvae typically lie in the back of the cell in the shape of the letter C. They're white, they're plump, they glisten, they look happy and healthy, and as they grow, they fill that cell. The bees cap over that cell, and then the larvae stand up in that cell and become prepupae. After a few days, the larva molts into a pupae, and you get the pupal stage of development under that capped cell. So how do individuals infected with American foulbrood present as they age? Well, they look mostly normal in their larval stage. In fact, you don't usually see it causing illness in the larvae. They'll develop seemingly normal. Maybe if it's going to kill them late in their larval development, they'll start to get discolored. They may twist a little bit in their cells. And when I say discolored, I mean orange, brown, black, coffee colored, that whole kind of spectrum of colors that look like essentially decaying larvae. But the real clinical signs typically manifest in capped cells. So in the standard cell capping of a worker cell, you get this nice dome capping on top of the cell. Well, with American foulbrood infected individuals, those cappings will become sunken. And not only will they be sunken, you might see perforations in those cappings. And if you were to open up or remove that capping and look into the cell, the individual in that cell is not plump. It's not white. Its body's not differentiating appropriately to where you see the head and the thorax, etc. What you get is an individual that really has a molten appearance. They're discolored, they're misshaped, and if you think about cell geography, so I'm looking into the cell from the camera side. If you think about cell geography, that individual developing in the cell will start to sink to the bottom of the cell. So this surface is the bottom of the cell, not the part that the queen laid the egg on. That's the back of the cell. So you'll get this individual kind of slowly becoming discolored and molten to the bottom of that cell. And once they are molten at the bottom of that cell, they form a very thick, sticky, firm scale. And that scale is incredibly important to know when it comes to colony illness with American foulbrood. European foulbrood also causes individuals to form a scale on the bottom of that cell. but you can remove that scale really easily from the cell. Whereas the American Foulbrood caused scale becomes almost fused with the cell wall, so to remove that scale, you're going to damage the cell. In fact, one of the key diagnostics for American Foulbrood is looking for that scale. It doesn't just have to occur in cells that have those sunken perforated cappings, because the bees will go into those same cells, open up the capping, and try to remove the cell contents, but be unable to do so. So you can find those scales in open cells and in closed cells. So I want to show you how best to do that using a standard frame. Again, the geography of a frame is such that the bottom of the cell is that surface. So that's where you're typically going to see scales. It does you no good to hold a frame and look into cells straight on. The scales aren't in the back. They are in the bottom. So really the best way to look into a cell for an American Foulbrood caused scale is to stand such that the sun is over your shoulder, and then to take the comb and tilt it to where you see the bottoms of all the cells. And so now I can see the bottom of all of these cells, and I'm looking for scales that have formed on the bottom of cells. Incidentally, you're going to find out shortly that's one of the really good ways to prevent bringing American Foulbrood into your apiary in the first place. But hold on to that idea. So I'm looking for these scales, I'm looking for sunken cappings, I'm looking for perforated cappings, I'm looking for individuals that appear molten, discolored, maybe disshaped. Another thing that you'll look for when you think about American Foulbrood is in some cases, as that individual starts to melt to the bottom of the cell, a condition called the pupal tongue. So that developing pupae has a proboscis that can get stuck on the roof of the cell. And as that individual just kind of melts away from the roof of the cell, you'll get this classic, what we call a pupal tongue sticking up. Actually, the pupal tongue is fairly diagnostic for American Foulbrood. If you see it, there's a strong probability that's what's killing the individual. But the pupal tongue is not a very common thing. You might see it once in a colony or twice in an American Foulbrood infected colony. So while it's pretty diagnostic, it's not overly common, but it is important to know that. So again, to recap, sunken cappings, perforated cappings, scales forming on the bottom of cells. When you open up those cells, discolored, misshapen individuals. Oftentimes when beekeepers see that sunken capping with those perforations in it, they will open up that cell and use a matchstick or a small stick, put into that cell, stir the cell contents, and slowly remove that matchstick or that twig. What they're doing is they're looking for the rope test or the ropiness of the individual that died in that cell. So for example, if you stir it up and pull out that stick or that matchstick slowly and no rope comes with it, then it's less likely to be caused by American Foulbrood bacterium. On the other hand, if you stir the contents and you slowly pull out that stick or that matchstick and you get this long rope, stringy, snotty looking rope that slimes out of the cell with the stick that goes longer than a centimeter, that's another particular thing that beekeepers look for when they're trying to diagnose American Foulbrood in their colonies. So 2 more things that I want to point out when you're trying to diagnose American Foulbrood in your hives. Number 1, foul. Number 2, brood. So foul brood means that you might have a smell associated with it, and brood means it kills brood. I've talked about all of that. Well, if you've got dying brood, you're going to have spotty brood patterns. So do you smell a rotten smell? Do you see a spotty brood pattern in tandem with all of these other things? Now, everything that I've just described, pieces of those different conditions could be confused for other things, sac brood, European Foulbrood, other conditions. But if the colony's displaying all of those things, there's a strong probability that's American Foulbrood. There are 2 other ways that you can have this particular pathogen diagnosed in your colonies. Number 1, they make a lateral flow device that you can have with you that you take in the field. The lateral flow device is specific to American Foulbrood. It's very similar technologically to a pregnancy test, where you take out what you believe to be an infected individual, you put it in a small jar and shake it with a liquid, you take that liquid off, put it on the lateral flow device, and that liquid flows through the device, and you get a banding pattern that's unique to American Foulbrood. There are lateral flow devices also for European Foulbrood. So I recommend that every beekeeper who goes into the field actually have that lateral flow device for European Foulbrood and the lateral flow device for American Foulbrood in their standard apiary bucket or pail or whatever they take out to the field, so that those things are always available to help you look at brood in the event you believe you have a brood problem. The third way that you can have American Foulbrood diagnosed in your colonies is you can work with your local apiary inspector or your regional, national, international diagnostic lab that will perform diagnostic services, typically using things like microscopy or polymerase chain reaction PCR to determine what it is that you have. If you go through any one of those diagnostic series, either the field series or using the lateral flow device or sending your sample off for positive identification, if you go through any of those and it comes back positive, there's really only one good option for your colonies. Think back to the background, the very introduction of this particular topic where I said Paenibacillus, the bacterium that causes American Foulbrood in colonies, has these two stages. The vegetative state that kills the bees, it's the thing that caused all of the conditions that I just told you about earlier, and the spore state. It's conceivably possible to treat your colony with antibiotics and kill all the vegetative individuals. The signs of infection will go away. You won't get clinical signs. The colony looks like it's made a rebound. The only problem is you have those dormant spores persisting in the woodenware, in the combs, in the honey, waiting to reactivate, as it were, downstream. The point that I'm making is once a colony has American Foulbrood, it always has American Foulbrood, which is why here in the U.S., multiple states consider this a reportable disease. Countries around the world, a lot of them consider it a reportable disease where you actually have to notify someone about it. That individual, often an apiary inspector or someone like that, maybe a veterinarian, will come out and take steps to help you get rid of that colony. Here in the U.S., even if it's not notifiable in your state, you are strongly recommended to dig a pit, put the colony in it, and burn the whole colony. The lid, the boxes, the combs, euthanizing the bees, and everything. It sounds terrible. It hurts my heart to tell you that, but that's really the best way to deal with that disease, and that's what limits the spread. Again, you cannot rescue a colony from American Foulbrood. Once it's there, it's there. So the best way to deal with American Foulbrood is to burn the entire colony and just try to do away with it. You burn it in the pit, you cover it up with dirt, and that's that. So euthanizing the bees, destroying that equipment is the best way to deal with it. Now you've got this other issue. What about all the other colonies in the apiary? So I would look at those colonies closely to see if they're showing any clinical signs of American Foulbrood. If they don't have clinical signs, that's good. So you're not having to worry about having to burn any of those colonies. If, on the other hand, you find colonies with clinical signs, you've got to rinse, wash, repeat. You've got to take those colonies, burn them, their woodenware, etc., and bury them in a pit through this whole process. But let's default back to the first possibility. You've burned the one, you've covered up the ashes that you knew to have American Foulbrood. Now you've gone through the rest of your apiary and you've not found the disease. What can you do? Well, here in the U.S., oftentimes people will work with their veterinarian to get a veterinary feed directive so that they can purchase and use antibiotics to treat those colonies that were in close proximity to the one that died. A lot of beekeepers around the world don't have access to antibiotics to use in their colonies because it may be illegal to do that. So what do you do in those cases? You monitor closely those colonies, specifically the ones that were in close proximity to the one that you had to euthanize and burn. You sanitize your hive tool. If you use gloves, you sanitize your gloves. You sanitize your smoker. You sanitize any equipment that you are using and sharing between all of the hives in your apiary. You watch that apiary closely and don't move colonies in and out of it. You don't exchange frames of bees or brood or supers of bees or honey within this apiary. You don't harvest honey from this apiary to feed the bees at other apiaries. You really want to almost quarantine this apiary and watch it for 6 months or a year to make sure there are no other colonies that show these clinical signs of infection with American Foulbrood. So really the best way to deal with American Foulbrood is to never get it in the first place, which is what leads to this last series of recommendations that I'll make to you, which are recommendations all related to prevention. Use bees on new combs, new boxes, right? New frames. This idea of using new equipment, new bees, hopefully prevents you from having the problem in the first place. Again, watch out about moving bees and brood between apiaries. You don't really want to share bees and brood, especially if you have reason to believe that American Foulbrood led to the demise of one of your colonies earlier. And I want to go back to a point I made earlier about looking for scales in the combs. If you purchase nukes or full-size colonies, you can inspect those bees before they leave the individual's apiary from whom you purchased the colonies. And what are you inspecting for? You're inspecting for all of those clinical signs I've discussed, but most importantly, for the scales that form at the bottom of those cells in the frames. You can pick up those frames, as I've mentioned. You can look in the bottom of the cells for those scales, and if you see scales of any sort, you don't purchase those bees and bring them back to your apiary. So the last thing I'll tell you when considering prevention, in addition to new boxes and all of that stuff, is to use hygienic stock. Honeybees can have enhanced hygienic behavior, which is the trait where workers in the nest detect diseased or dying brood. They'll uncap the cell containing that diseased or dying brood, abort that individual, and clean up the cell prior to it receiving an egg from the queen later. So bees that have high levels of hygienic behavior or hygienic expression tend to have low levels or negligible levels or no levels of American Foulbrood in the nest. So I always recommend, in addition to starting with new equipment and putting just bees into that new equipment, that you use a hygienic stock in your apiary, because she's likely to lead bees that keep that disease well below clinical manifestation. American Foulbrood is a terrible disease. It's one of those things that created apiary inspection programs around the world. The best way to deal with it is to not get it in the first place. But don't consider yourself a bad beekeeper if you happen to see it in your apiaries. American Foulbrood spores are everywhere. If you keep bees long enough, at some point you're going to have to deal with it in your apiary. It doesn't mean that you're a bad beekeeper. It could mean that there were robbing bees, your bees are robbing another apiary or a diseased colony, etc. There's lots of reasons that your colonies can get it. But if you see it, it's important to deal with it quickly, efficiently, and directly to limit its spread. And if you're able to do that, then you can keep the disease at bay. Then you'll have many, many decades that are happy decades, both for you and for your bees.